Well, welcome back, everybody. So now that you've learned how to do Mendelian genetic problems, I thought I should go back and give you a little bit of the history on what Gregor Mendel did and why it was so important. So let's get started. Who was Gregor Mendel? Well, he was an Austrian monk who lived from 1822 to 1884, and he had a fondness for pea plants. And the reason he liked pea plants was because they were cheap, they were easy to obtain, and they could either self-pollinate or cross-pollinate. And so he wanted them as sort of a model experimental system. He knew what he was doing in a sense. He knew he was going to find something interesting scientifically. And this was a very easy system to work with. In addition, he developed populations of pea plants with paired traits, and I'm going to show you those in the next slide. And he would hand pollinate plants with alternate traits to see what the offspring looked like. And here in the background you can see a pea plant, and here's Gregor Mendel himself. Now, here are the traits he used. So he had 7 different traits, as you can see: flower color, plant height, seed color and shape, pod color and shape, and flower position. And each of them had 2 alleles. One was a dominant allele and one was a recessive allele. What he would do is he would grow populations with each of these traits until he was sure that that population was true breeding. And what that meant is if he crossed any 2 with each other, he would always get offspring that had the same traits, so they bred true. And what we now know that means is that the plants were homozygous, whether they were purple flowered and homozygous dominant or white flowered and homozygous recessive. It turns out that you can use a brush, a simple little paintbrush, and transfer pollen from the anther, which is the male part of the flower, to the stigma, the female part of the flower, and cause fertilization. And this is what Mendel would do, is he would take pollen from one plant with one set of traits and put it onto the stigma of another plant with a different set of traits. So this means you can know the parents of the offspring and then grow them out to see what their traits are. So it was a beautiful system. And if you clip the anthers, as being shown over here, then the plant cannot self-pollinate. And so that prevents confusion in your results. In Mendel's experiments, here's what he did. He would take the parental generation of 2 different true breeding plants that exhibited alternate traits. So in this example here, a purple flowered and a white flowered individuals, and he would cross them to produce the F1 generation. He would then allow the F1 generation to mate with each other, and actually most of the time he just allowed them to self-pollinate to produce the F2 generation. And what he found was that the F1 generation always exhibited the dominant trait, and that in the F2 generation, the recessive phenotype reappeared, but it was always about 25% of the offspring. In other words, the offspring of the F2 generation were 3 to 1 dominant to recessive phenotypes. So let's make sense of what happened in Mendel's results. And I'm sure you can already see this because you're already experts on solving genetics problems. But in the first generation, he crossed 2 homozygous parents. In the flower color case, big P by little p, little p. And this meant that the entire F1 generation was heterozygous, big P, little p. That's pretty straightforward and simple. Now don't forget, Mendel had no idea of what a gene was, much less what an allele was at the time. So this was very interesting and surprising results. He actually thought the crosses would produce intermediate phenotypes, which if he had chosen codominant alleles to work with as opposed to dominant recessive would have been the result, and he might never have published his results. So all the F1 then, being heterozygous, displayed the dominant phenotype. In the F2 generation, he crossed 2 heterozygotes, a big B little b by a big B little b. And as a result, as you can see in this Punnett table, 1/4 of the offspring are predicted to have the recessive phenotype, and that's what Mendel found. You will often hear these types of genetics problems referred to as the monohybrid cross. And so why is that? Well, because the parental generation was homozygous and true breeding, the F1 generation is often referred to as a hybrid between the 2 true breeding strains. And the F1 generation doesn't breed true. It itself is purple, and it produces both purple offspring and white offspring, so it does not breed true. Therefore, breeding the two heterozygotes with each other is seen as breeding the hybrids. And since only one trait is involved, it is the monohybrid cross. And you can now guess what the dihybrid cross would refer to. If you don't understand it, make sure you bring that question up in class. Technically, only when the two heterozygotes are crossed should we call these problems monohybrid crosses. Now, before I go on, I just want to do a quick aside about the progress of science. Mendel published his results in the early 1860s, just a little bit after Darwin published his ideas about evolution. But Mendel's results remained unknown until about 1905, when geneticists both in Europe and the United States discovered his paper and immediately realized the importance of the data. Darwin went to his grave believing that inheritance was blended, meaning that when two opposite traits were crossed, the offspring would be intermediate. And he worried that this meant that when a new mutation showed up, that the offspring all would only show it partially, and therefore there might not be a benefit. And he went to his grave not understanding what Mendel had said, even though Mendel was a contemporary of Darwin. So, what you should know from all this is as follows. First, you should understand the experimental design that Mendel used, the true breeding parental generation, the F1s that were made from that, and then self-pollinating or breeding the F1s among themselves to produce the F2 with the famous 3-to-1 ratio of phenotypes. Second, you should know the genotypes and phenotypes of each ratio. You should just be able to describe that without any prompting whatsoever. And finally, you should know the following vocabulary: true breeding, parental, F1 and F2 generation and what they mean, and monohybrid and dihybrid cross. That's it for today. Until next time, go out into the field, look at nature, ask biological questions, and bring them back to class. And I'll see you soon.